Hi, Steve. Good to see you.
Great.
Okay. I think we should start. And if anyone else joins, they're welcome. I think we have enough people around. Maybe we should start if you're all okay with a quick round of introduction. Uh, I'm not going to introduce myself. You all know me, but Steve, we're very excited to have you here and appreciate your time to join us on the call today and talk to us about the in vivo models subcommittee, which is wonderful. Um, Maybe we start with you giving us a very quick introduction about who you are, and then we'll go around the room. And I would love for our patients and everybody on the call to introduce themselves. And then we would love to hear from you, Steve. How about that?
Yeah, I've got a little bit of an introduction already built into the talk as part of my story.
Oh,
So, how
perfect.
about I go last and then we can launch right into the talk with that?
Okay, that sounds great. Um, Lisa, I assume everybody knows you, but if you want to say anything, is in the beginning, would love to hear from you.
I'll just say hi.
Hello. Okay, why don't we go with Susan and Susan Gleason.
Hi, I'm Susan Gleason. I was diagnosed with PKD in 2008, incident to a hernia surgery, and had never heard of it before. So ever since then, I've been very interested in trying to keep up with all the research. So I really appreciate you being here tonight. Thank you.
That's great. Thanks, Susan. Julie.
Hi, I'm Julie Hogan. I live in Las Vegas. I was diagnosed with PKD when I was 26. My parents do not have it. They were tested. I recently reached my 5-year kidney transplant anniversary. I was a speech pathologist before, and I have become now a co-founder of a company where we help people get listed for kidney transplant. And so I'm a patient navigator along with that, trying to help people get through it.
That's great. Thanks, Julie. Corey.
I'm Corey Anderson. I'm a PKD patient navigator at KU. I was originally diagnosed with PKD, but have since taken a genetic test, and I have kind of some cystic renal disease that no one really knows what it is, but I'm treated as if I have PKD. I'm trying to think what else anyone else said, but other than that, because I work at KU, Dr. Parnell tells the best stories, so I think you guys are in for a good one.
Oh, that's great. And Susan Mendley, welcome. We're happy to have you on the call.
Thank you. I always panic when you say Susan, and then I wonder, is it me or is it Susan Gleason? There are not that many Susans I know, but thank you for letting me join you.
Of course.
Susan, you travel in the wrong circles. I know a zillion Susans.
And when I was in elementary school, I knew a zillion Susans too, but they're harder to find now.
Ah,
Okay, great. Well, thank you for being here. I know Vino is on the call.
Hi, everybody. Good evening. My name's Vinamrata Rao or Vinu. I am a third year MD PhD student, or third year in graduate school and fifth year but I'm working to wrap up my thesis and my dissertation pretty soon, but planning to continue working with patient engagement groups. So I'm really excited to read to hear what Dr. Parnell has to talk about with the in vivo model.
That's wonderful, and we're very excited for you, and we're excited about your decision to continue to work with the patient engagement group. That's wonderful. Taslima, I see that you joined us. We're just doing a quick round of introduction, and you would be the last one to introduce yourself before Steve.
Wow. Okay. Good evening, I would say. And I'm retired from my work, but like I just wanted to know more about PKD. So this is I am doing like and at the same time, it is not just learning what is going on with the PKD. Also, I'm thinking if I can use my experience, that would be great. So that's what it is.
It's wonderful. Thank you, Taslima, for being here. And Nicole Reed, which I think we all know.
Hi.
Hi, Nicole. Thank you for being here and for keeping us on track all the time.
I try to do the best I can.
You sure do. Thank you so much.
Thank
Okay,
you.
Steve, we hand it to you.
All right, thank you. And thank you all for coming tonight and participating in this group. It's been a real asset, I think, to the RRC to have the patient engagement. And as long as you don't have a lot of noise in the background, you can feel free to leave your sound off. I'll be asking you some questions and make this a little bit interactive to get your feedback from things. And I also recognize a number of you. Corey, I know that I know you. Susan, I feel like we have met before. I'm about 95% certain. And Julie, I think you as well, maybe at one of the meetings out in Colorado or PKD Connect or someplace like that. So let me start a little bit with my story, because my story, if I can get it to work here, my story is really your story. This is my grandfather right here, and his sons, my two uncles, and my father right here. Every single one in this photo has PKD. My grandfather passed away in 1963 with his kidneys looking like this. This was shortly before dialysis became available. So when his kidneys failed, that was it. That was the end of his life. There was nothing that could be done for him. This uncle here received a transplant and lived 20 years with it and died of causes unrelated to PKD, did very well. This uncle died about age 50 or so, I'm convinced, from complications, probably some secondary complications related to PKD. And this is my father. He also received a transplant and lived for about 20 years with that and died of complications unrelated to PKD. So I have grandfather, father, uncles, siblings, nieces, and nephews, all with PKD. And it's really been remarkable to see the change from my grandfather, who died with no treatment, to now we're at a place where we have at least a treatment and we're working on better treatments. So it's encouraging to see, what the future holds. So again, your story is my story. I come from a PKD family. I don't have it myself, but I understand what you're dealing with, and I have a lot of interactions with my family and have to communicate what I know. I've been blessed to work within the PKD field. How I got into the field beyond the family relation is another story for another time. But I just show I'm a real person. In my free time, I like to climb mountains, and I take my favorite PKD shirt, which is a free gift from the PKD Foundation. I love this shirt. I like getting it to remote locations and getting photos to snap to try to raise awareness for PKD. So that's who I am. I am a biochemist, a scientist, and I want to give a little bit of brief background in terms for some things that I'll be saying a lot of today. I've been told you really want to understand what we're doing in the in vivo subcommittee, and how does that help you as patients? So just a few terms for background. PKD, unfortunately, you all know what this stands for. That's why you're here in the first place, autosomal dominant polycystic kidney disease. You hear me talk a lot about proteins, and if you're in the biological sciences, we're very concerned with proteins. I'm not talking about a hunk of steak or a piece of chicken breast, but proteins are the names for the tiny biological engines that live within our bodies, within our cells, and they make everything inside of us work. Just about every sort of process that you have out there are proteins for that. Be it the way that your blood clots when you get a cut, be it what tells your cells to proliferate and divide and make more cells, be it to tell the cells to stop proliferating and dividing. We have proteins that build things, proteins that tear things apart. And, we have proteins that make our kidneys do what they're supposed to do. Proteins are encoded within genes. Genes are within our DNA. That's our genetic material, what you inherit from your mother and your father that help make you who you are. And mutations in these genes, like PKD1 or PKD2, can lead to mutations in how these proteins will work. One thing which I've found that who aren't in the sciences often misunderstand is the genes, many genes are named after diseases. And so they tend to think that, well, I got some gene that nobody else has. Well, that's not true. We all have PKD1 and PKD2 genes. I have a PKD1 gene and a PKD2 gene. I actually have two copies of it, one from my mom and one from my dad. What's different about me than from you is that the copies that I got from my mom and my dad were normal copies, whereas you picked up a mutant form of the gene, and that's why you have ADPKD. So here's a little dirty secret within our field, not so much of a secret. Scientists, as biologists, molecular biologists, we have the ability to introduce mutations and other changes to these genes to see how these changes will affect how proteins function. So you can do this in a test tube. This is called in vitro, or it can be done in a living organism. And if you do these types of experiments in a living organism, this is called the in vivo. And that's what I do. I'm part of the in vivo models subcommittee. Well, why would you do things in vitro versus in vivo? Yeah, Reem, did you have a question here?
I actually wanted to ask and didn't want
Yes.
to interrupt you, Steve. So if you're okay, I would love to record this because we have some members of our team who couldn't make it today.
Yeah, absolutely.
Okay, we'll make that available for them if they would like it. So I don't know, Nicole, if it should be you who you record.
Yes, it's a recording.
Okay, perfect. Thank you.
Great. Yeah, that's fine. So in vitro work is important, easier to do, but it can be a bit misleading. Just to give you an example of this is an analogy. Analogies break down at some point, but if you were to take a man and stick him in a room and the only food supply that you were to give him was rutabagas and haggis, the sheep stomach sausage, eventually that man would get hungry and eat it, right? And so you could conclude from that the man can eat rutabagas and haggis and he could survive on that. But now in vivo work is important. Harder to do, but it's more representative of biological reality. So let's go back to that experiment with that man. You take that man, you put him out into the real world, he's probably never going to eat rutabagas and haggis. That's just a fact. So that's how these two things are a little bit different. But I live in that realm of the in vivo model subcommittee. We're doing things in actual living organisms. And now we're going to drift into some wonky slides, and we'll drift in and out of this, and then we'll come back to analogies to help explain in lay terms what we're doing. But this is a line for what we do. We have a mission to ensure access to animal-based resources. The goal is to accelerate the pace of research into pathogenic mechanisms of PKD and possible therapies by serving as a centralized hub for the generation and distribution of genetically engineered zebrafish, mouse, PKD models, novel reporter lines, and PKD-related biomaterials. We've created two mouse lines with tagged PKD proteins, and don't worry, I'll explain what it means when we tag a protein, to allow researchers to study the function in vivo. Again, that's inside the actual animal. So we're generating mice that contain patient variants of PKD2 to study channel function in vivo. And we also have a biobank that provides tissue samples to researchers to help move their projects forward without the need to generate the actual animal models themselves. And here are, again, some wonky things down here. And if you're a biochemist, then this is how we represent proteins to help us get some idea of what they look like and what they're doing. We'll take another step back here. We're an in vivo subcommittee, so we're using live animals. So a lot of people ask, well, why do we use animals? And let's be clear, most of the work that we do is in mice. So remember that the in vivo work is important. It's representative of biological reality if we can actually get into an animal system. Mice have many of the same organ systems as humans, including the kidneys and the whole system of filtering your blood, filtering toxins out of your blood, and peeing it out in the form of urine. Mice also have PKD1 and PKD2 genes, just like humans, very, very similar to ours. And if you mutate the mouse PKD1 and the PKD2 genes, then it leads to polycystic kidney disease, almost just like in humans. There's some subtleties that I won't get into unless you really want me to, but suffice to say, we can introduce PKD into mice, and then we can do studies in those animals because mice are inexpensive to maintain. They can produce a lot of progeny, that's a lot of offspring. So we can also change the genes in mice fairly easily. So what I tell people that I do is that I make mice, and I make mice that have variations in the PKD1 and the PKD2 genes, and we do this to try to help figure out how and why the PKD genes work, what goes wrong in the mice with these PKD mutations, and also how we can better treat ADPKD. So now I'm going to take you into some of the nuts and bolts of what the three different committee members are doing. The committee consists of ourselves at KU Med, and then University of Alabama, Birmingham, and also University of Maryland. This is a project that was taken on by my colleagues at UAB in Alabama. And what they've done is they've generated the Polycystin-2 Halo mouse model. Well, Polycystin-2 is one of the proteins that's defective in people with ADPKD. So that's why we're working on this particular protein right here. I'll tell you more about what tagging means. And this is how, as a scientist, we represent the look of proteins. And basically what I'm showing you is all the different parts of the protein. Just like if you have anything, like you have a car, and we'll talk more about cars later, you've got different parts to the car. Well, these are just the different parts to the protein. And what we've done here is we've stuck this tag on here, this so-called halo tag, because proteins are a little bit modular. You can add bits and pieces to them, and they'll continue to function in the context of a protein. It's sort of like if you have a car or a van, and you attach a trailer to the back of your van. So now wherever your van goes, the functionality of that trailer travels around with the car. So you can use it wherever you take your van. Same thing with these tags. We'll put a tag on that protein that has certain properties to it that are useful to us. And in this case, it's to visualize the protein, to see where it goes within the body. Well, what good does that do us? Well, I'll explain that by refreshing you of a children's book, Where's Waldo? I trust most of you are familiar with Where's Waldo. So in Waldo, we have our the little cartoon image of Waldo, and he's got this red and white striped shirt, and he shows up in these artist renditions where there's all kinds of other red and white striped things, and it's difficult to find Waldo. If you're looking at this, if you got a real sharp eye, maybe you've picked him out, but I'm going to guess most of you are still busily hunting for Waldo. Well, let me give you a little bit of help here. Now, can you see him? Here he is right there. This is the same type of thing that we do when we talk about tagging a protein in biology. We stick this big fluorescent green tag or different color tag on our protein, and then we can see, just like you can see Waldo here, you can see where he is much easier in this environment of confusion. And that's helpful because you can tell from that, oh, Waldo lives in this particular city or this particular town. He's located right here. But more than that, because we're dealing with in vivo stuff, this is stuff that's alive. So these proteins are active and they're dynamic. They move within a cell. So if you can imagine, you can trace where Waldo goes in this environment. You can easily see just by tracing this fluorescent tag, he's moved himself over this particular building right here. So let me ask you now, let's definitely take your mics off now. Let's do some experiments. Let's hypothesize, why is it that Waldo is traveling over to this building every day about the same time? Go ahead. What do you think? Why is Waldo moving over to this building? Cut out your ideas.
He wants to have some coffee?
Okay, he wants to have some coffee. Any other ideas?
Meeting a friend.
Meeting a friend? Okay, great. Let's work with that. And I'll give you one more. Maybe he's maybe he's a bit of an artist, a connoisseur of art, and we see we've got this little poster here, and he likes that poster, and every day he stops by to take a look at it. Let's say you're the mayor of this town. Let's say that you can perturb the environment here. What would you do if you wanted to test that Waldo is coming over here? Look at this poster that's posted up on this building. How would you test that?
Take it down.
Yeah, you take it down, you move it over here. And if you took that poster down, you moved it over here, and you follow this green tag, and all of a sudden Waldo goes over here, you know, aha, Waldo has some affinity for this poster here. He wants to see it, he wants to look at it, so he goes over here, and you can test that. Okay, well, what if Waldo goes here? Well, then your hypothesis is wrong. What about the coffee or maybe the food? What would you do if you wanted to test that?
You could move the coffee too.
I move the coffee. So it looks like you've got a little market center down here. So if you had these people set up a food cart of some kind, and all of a sudden, if you set up that food cart, all of a sudden, Waldo goes down here. Well, you say, oh, well, Waldo splits his time between here and here where the food is. That's why he's going there. Same thing if you wanted to test, what if he's just going there to meet a friend? Well, maybe you could trade the people working here. It looks like they're selling store. If you sent them up here to work in the cafe, and you took the cafe workers down here and had them selling plants, if Waldo went down here all the time now, you'd say, oh, well, Waldo has friends and he's just dropping by to visit his friends. Well, or what if Waldo just stopped showing up together when you did that sort of switch? Well, from that, you would say there's some sort of a compound effect. Waldo maybe likes the food and he likes the people, and he likes that environment in this particular place. When you move the people down here, no longer serving the food, so the food tastes awful up here, so he no longer comes, and he doesn't really like the people if they're not serving the food, so he just doesn't come to either location. You can see there's all sorts of ways in which you can perturb this system, and then you can use the fact that you can track where Waldo goes to understand what drives Waldo's behavior. Well, we're doing exactly the same thing with polysystem 2 in this scenario. See, we've stuck this tag on here, and in this experiment right here, in this case, the tag is a red color tag. And so we can see, oh, that polysystem 2 protein is showing up in the brain. We don't entirely understand why it's showing up in the brain, but that's something new that we learned from this experiment. And it's actually very, very low levels. We can hardly detect it in the kidney unless we take the kidney and break it up into individual cells. And in this experiment here, now where we're using a white tag on the protein, we see that it's present within the cells, and it's present at this part of the cell called the cilia, which people have been interested in for a long time. So you can see how the ability to track and follow where this protein is going is telling us a little bit about its behavior, and it's giving us new ideas and new things that we can test. So when people talk about tagging a protein, that's the goal. That's what they're trying to do. So I'll just pause there and ask if you have any questions before I proceed.
So then most of the researchers start with a hypothesis, and then they use the tag to test it. The tagged.
Yeah. So one hypothesis that you might test would be, okay, well, maybe if you subject the kidney to some form of renal injury, then it would make that protein show up. And so you could do that. You could subject the kidney to some kind of an injury in these mice, and then you can look and see for the appearance of that red marker that would pop up. And if it did, then it would say, aha, there's something about injury that's triggering the production of the polysystem 2 protein. And maybe it might go to a particular place within the kidney, and that would be telling us what part of the kidney is affected by that injury. So yeah, absolutely. Any other questions? All right, so we'll move on to work that was done here at KU Med. We had a similar idea. In this case, we wanted to add a tag, slightly different tag, but same overall function, to the polysystem 1 protein. Polysystem 1 is a product of the PKD1 gene, and most people with PKD have mutations in their PKD1 gene. So we wanted to see where the polysystem 1 protein is going. So we put that tag on there. And just like before, researchers can use a fluorescent ligands to track the movement of this protein. But we couldn't see the protein anywhere. Despite our best efforts, it simply wasn't showing up. And I won't get into reasons for that, but what I will tell you is something else happened. When we put that tag onto the polycystin 1 protein, we observed that mice with that tag started to get polycystic kidney disease, like you see here. These cysts here, just like you might see in a PKD patient, were showing up in mice. These mice are about 8 weeks old or so. So this was unexpected. We weren't expecting this. And we call this type of mutation hypomorphic. Now, typically, and don't worry, I'll explain that word more in just a little bit. If you were to delete the entire polycystin 1 protein, these mice wouldn't be born at all. It's essential for development. So this mutant form of polycystin, the mice are still born, their kidneys develop, they even look normal for just a little bit, but over time, they start to develop the hallmarks of polycystic kidney disease. So when I say hypomorphic, I mean it has some functionality of the original protein, but it's limited in some way. Let me give you another analogy. I've been doing car type analogies for a long time. I've actually found that most aspects of biochemistry can be explained with cars. And again, I want your involvement in this. So I'm going to tell you what the defect to your car is, and you're going to tell me what the consequence. And you're going to imagine that you're aliens from space that came down. You see all these cars moving about, and you have some sense that cars get things from one place to another, but you have no idea how the car actually works. And you want to understand what is it about the car that makes it function. So if you were to take and flatten that car with a steamroller, what's the consequence going to be?
It won't go.
Yeah, car does nothing. It won't start, it won't roll, it won't go. So, if this were a biological mutation, we would say it's null. It does absolutely nothing. Now that's helpful. It tells us a little bit, but it basically tells us that it's important, but it doesn't really tell you anything about the car, how it actually works, because you've just destroyed the whole thing. Okay, well, now let's talk about other smaller defects, like these so-called hypomorphic defects. What would happen if we broke the window on a car? What might you expect the consequence to be?
It would be colder on the inside. Colder on the inside, but it would still run.
Yeah, it would still run, but you might imagine maybe you drive a little bit slower out on the highway because of the wind disturbance. Maybe you wouldn't drive in the rain, or maybe you wouldn't drive in the colder temperatures or something like that. But you may go a long time before you ever really see how the defect correlates to an actual consequence. Not until you expose the car to that type of condition. Maybe it's the highway driving, maybe it's the rain, maybe it's the extreme temperature. Tells you a little bit. Well, what if you got a dead battery? What's the consequence now?
Won't go.
Won't go. What if you push it down? What if you take the brakes off and you push it down a hill? Does it go?
Maybe.
It'll roll down the hill. It'll roll under gravity, but it won't start. But now, see, you can you can bring a friend with another car. You can hook up cables between them, and you can get this car to work just fine just by jumping it. That's a different defect than the broken window. How about this? No gas. Is the car going to roll? If you push it under gravity, it's going to roll downhill, but it won't start. Even if you get help from another car, unless that person is bringing you a couple gallons of unleaded, that car isn't going anywhere. But it will still roll. What if you take the tires off? Can you start it? You can start it. The engine will turn over. The engine will go. The car's not going to go anywhere. It's not even going to roll downhill anymore. Now, what if you remove the brakes? Car start? Car starts. Car rolls. Car won't stop rolling. Okay? So start to put all of these things together, and you develop an understanding from this that, electrical system from the battery is somehow connected to the gas, which is somehow connected to the tires, and all these things put together makes the car go. And you could continue to take pieces off the car, be it the spark plugs or the starter or the fuel pump, and you would understand after a while all those things make the car work, and you'd also understand the brakes is an important component of that. The car goes, but you need to have the brakes to make it stop. So these are hypomorphic mutations if we were to express this in terms of biology. And that's exactly what we're seeing in this particular animal right here. This is a hypomorphic mutation. We still have function, like if it's a car, it's still rolling. Maybe you could start it, but it's got some little defect to it. But we don't know what the trigger is. But if we could figure out what that is, it would help tell us something about what the function of this protein is. In terms of how this is affecting our understanding within the lab, remember I told you that proteins are made up of all these little bitty parts to them, and those different parts have different functions. Well, we're taking a closer look at the particular spot where we put this mutation, because where we inserted this tag, because the tag probably interfered with an important function of the protein. And if we could understand what that function is, then it might give us some clue as to ways to trigger this protein to function the way it's supposed to for patients with PKD. Okay. Any other questions about that aspect of what we do? All right, great. So now we'll move on to the work of Maryland. In this case, this is sort of like an extension of our analysis where we're taking little bits and pieces off of a car to see what it does. But in this case, we're approaching it from a different angle. In this case, we know what the polycystin 2 protein looks like. This is what it would actually look like if you were a little like the size of a subatomic particle and could see the protein with your own eyes. This is kind of what it looks like here. And the different letters and numbers here are just different positions and places on the map of the protein. And this is a part of the protein that we think is very important based on where it is within the protein. We think it's involved in a critical function. And we know in patients, there are a number of patients who have mutations at this particular spot within the protein. In this case, my colleagues in Maryland mutated the protein at this particular site, making the prediction that it would give these mice polycystic kidney disease. And that's exactly what we see in the images here. This is what pups very early during gestation look like if they have mutant polycystin proteins. They actually aren't born. You don't get to see them at the state where they have cystic kidney disease unless you do some other types of tricks of the trade that I won't get into. That's over here, and you see that this mouse has the cystic kidneys like you see over here. So this is useful because this is helping us to study a particular part of the protein to make predictions about those areas that are functional. And this will facilitate the development of really personalized medicine approaches for ADPKD patients, because you might imagine that the therapy for a person with this particular mutation or mutations that are somehow linked to this, just like with the car, we had a linkage between the battery to the gas, to the transmission, to the tires. This might be able to help us understand how the different parts of the protein will consolidate about this particular function right here in order to develop therapies for you, the patient. So the final thing that all of us contribute to is this biobank. And the biobank is a repository of kidney sections and pieces that we take from these animals that we've made. So we do this because we'd like to see more people come into the field, more people with different ideas about the disease. And often within the sciences, you might have somebody who's working on something that seems to be completely unrelated to polycystic kidney disease, but it may be something that they observe makes them think, there's some kind of a link here. I wish I could test that. Well, to test that, it's very helpful to have these biospecimens that come from these animals. But if you didn't have any experience in this field and you wanted to get into it just on your own, it would probably cost you about $10,000 to $15,000 and a year to a year and a half to get all of the different reagents that we have on hand within our cores in your lab, just to do a simple experiment that tells you, yes, there's something here, or no, there's not. So one of the things that we do for other investigators is we harvest these tissues, and when they call us up and they say, hey, I'd like to test some idea, I'd like to see if my protein is somehow linked to this pathway, we can send them those samples that have been preserved. They can do their experiment and they can get an answer rapidly without having to spend thousands and thousands of dollars and all that time just to get the material into their hands in the first place. So this is a way that the funding that's provided for the RRC is able to help other people within the broader field of science get better access to studying the disease. And again, the goal is to accelerate the pace of discovery and to make these reagents to the field to help you, the patients. So that's the conclusion of my talk. Just some images from some of my hiking excursions, just to have something to look at as we end our time here. And again, I'm happy to take questions, be it questions about science, be it questions about, you know, my side of the story, my perspectives on PKD from a patient family perspective. Really, whatever you want to talk about, I'm game. Yeah, Susan.
That was a great talk. Thank you very much. Just logistically, so if I'm a researcher and I call you up and I say I want to test this theory, like,
Yep.
How do you decide? I mean, do you just give it to me, or do I have to explain what my thought process is, and how do you monitor what I'm doing with it and get my
Right.
results?
Well, you know, we try to be as open as we can for people to test ideas. It's helpful for us to hear what the idea is and to respond to them. It may well be that they have very little experience in the field compared to myself. I've been in the field since 1995. So they maybe have missed some important literature from the past. Maybe somebody's already done this and we can say, hey, this has already been done. You need to read these papers and then decide if you really want to commit your time and effort to this. Maybe we know somebody who's already working in that area and we can put them in contact with another investigator. But for the most part, you know, if it's a new idea, we want to make the material accessible to them. You know, if it's a harebrained idea and we're super concerned, you know, the supplies are limited. You know, it does cost money and effort to generate the supplies. So I suppose there is somewhat of a barrier that we want to be sure that the people have really thought about their ideas and aren't just wasting the material, that they can do the experiments. But you know, most people have done their homework. They have a good idea. They come to us, and we're happy to provide that tissue to them.
And then, do you keep track of? Do they report to you? I found this, or I found this out. Or...
Well, we ask them to reference the core. That helps us to track and show that this resource that has been provided by the NIH has been a benefit to other researchers. We ask them to include in the publications acknowledgement of what they've done. And sure, we keep track of the material that we've been given to other people. And we really want to see it come into publication. Yeah, Taslima.
I have like a little different question. So as you mentioned that you are working on this area almost 20 plus years. So question is that how do you see, like looking at the research, it seems that it is very early stage of learning about PKD. So to find out a cure, it will probably take another 20 some years. So do you think that way, or you think you have a more positive idea that, oh, okay, no, like the solution, because it is started with mouse, and so it will be sometimes in the human being, the study. So that's why I said that it is another 20 some years to find out a cure.
I don't think so. And Lisa, I want to cue you up and get your perspectives on this. You might be gearing up your response to this as well. I don't think it will take that long because, for one, the pace of discovery has accelerated. If you go back to when I started in the field, we had just discovered those genes that encoded the proteins that I talked about so much today. And it probably took us a good, I don't know, maybe 10 to 15 years to get some sense of what they were doing. And to this day, we don't have a complete understanding of that. But the tools and resources that are available and our ability now to actually have some idea of what the structure of the protein looks like, that's new within the past 5 to 10 years, is really accelerating the pace towards a cure. We do have therapy with tolvaptan. There's a mechanistic reason why tolvaptan is effective, but we're getting more and more details about the function of the polycystin proteins, and even recent work has shown that if we can restore that function, that it antagonizes disease. So, from my perspective as someone who's very interested in the proteins, it's been a bit of a renaissance of interest in trying to get back to the fundamentals of what those proteins are doing. And I think if we could figure out what they're doing, then we have the inside track towards really antagonizing these diseases. Lisa, I'll let you put in your 2 cents on this.
So thanks, Steve. But before I do, I just want to tell you, I loved your car metaphors. You are obviously, I wrote this as a private note to you, but I'm going to say it to the group because you've given me an opportunity. You're obviously a very talented teacher, and we so appreciate your making this so available. To your question to me, I think that I would say 3 things. First of all, science sometimes is just a grind. It's just grinding it out. And there are lots of people doing that kind of work. And sometimes that just pays off. There's that one insight that is a tipping point and opens everything up. People are like, oh, well, this connects this to that. And there's a new understanding because there's a tipping point. And we only get to that tipping point because people are doggedly persistent along a line of inquiry. That's the first thing. The second thing is that that science is not a linear pursuit. I mean, sometimes there are just unexpected insights. I mean, I would remind us all that at least this is the lore that that Fleming discovered penicillin not because of dogged pursuit, but because he was a sloppy scientist. And but he was observant, and so his lab was a bit of a mess, at least according to the lore. And he was working with bacteria, and his plates got a little contaminated. But he was observant, and he noticed that where there was mold growing in the plates in his lab, that the bacteria wouldn't grow. And that was like, whoa, who ever thought of this? And that was the discovery of antibiotics. And I think the third thing that is really important is, and I'm not being gratuitous here, I think sometimes science moves forward in really productive directions because of these kinds of exchanges. People saying to investigators, this is important to me, and it kind of focuses inquiry in directions that, left to our own devices, we might not go in. So I think that there are a multitude of sort of approaches that move the scientific field forward, and these kinds of dialogues is part of that. I guess the only other thing that I would add to this, and it picks up on Steve's point about an explosion in science, and here I'd point out 2 things. One is we always thought that the course of PKD is inexorable. In other words, once it starts, it's like, I'm going to use a different metaphor, it's like a horse getting out of the barn, and it just goes. And there's nothing you can do to get that horse back into the barn. And then there was, again, work with mice. You can't do this with humans. Work with mice from Steve Somlow's lab that said, hold on, maybe some of these processes, you can not only slow them down, but you could actually reverse them. Now, there's a long way between those mice in Steve Somlow's lab and the clinic, but it opened up a whole area that we just never thought was possible. And then the companion to that is that we have heard about since the 90s gene therapy. But now we're getting to the point in terms of understanding fundamental biology that we don't have to necessarily put in the normal copy of a gene. We can kind of be a copy editor and go in and say, oh, this is misspelled. We're going to change that misspelling. And that has made therapy for a lot of diseases, not just functional diseases, but perhaps even structural diseases, possible to think about in terms of intervention. So the fact dogged pursuit is important, lots of people working on PKD. The fact that serendipity plays a role, the fact that patient dialogue helps focus us, the fact that that there are unexpected perspectives that come from animal studies, and the fact that we are learning about, for single gene disorders, how to edit the genetic information. I think all of these things bode well for an unexpected acceleration in the field. So Taslima, I think you're cautious to say 20 years, but I suspect that for some of the things that we're interested in, it's going to be much sooner. And the very, very final point, and thank you, Steve, for giving me the opportunity, is I think about some of the conversations we've had in this meeting and some of the folks that have really suffered significantly with this disease. I mean, Steve.
We are making the mouse strains available to other researchers, and we're hoping and counting on the wealth of different perspectives that people in other areas of PKD research or even other areas of science will be able to take those and use them for their own experiments. There are lots of different things you can test. Like if you want to talk about that SNAP PC1 that didn't behave the way we predicted, someone could approach that from trying to understand, well, why can't you see it? Maybe it's being cut up in some way during the processing of the protein, how it's made. They can address that question, or maybe they can approach it from a completely different question of, well, why is it that it's cystic? I know in my laboratory, I've actually taken that mouse and crossed it to other hypomorphs to see how they behave. And they actually, the mixing of the hypomorphs, they don't get cystic disease. So that's kind of a case like going back to the car analogy where you got a dead battery on this car and this other car here that's come in and jumped it in some way. That's an intellectual link there that can be made. And I'm just one of many who can make intellectual ideas and guesses and hypotheses. So we want to get these things into the hands of other people so they can test their own ideas.
Thank you. Other questions, thoughts?
I think I'm mixing up my memories, and I don't know if I have a question in here, but I vaguely recall at some science roundtable, somebody commented that rats don't naturally develop polycystic kidney disease, but pigs do, but pigs are expensive. Is that, what are your, is that even true, or what are your thoughts on that?
Well, remember how at the beginning I said that the mice get PKD almost just like humans do, but not quite. So you're asking me to go down that rabbit hole a little bit. So for humans, all you need is one bad gene and you've got the disease. We call that autosomal dominant. In mice, if you give them one bad copy of the gene, they're perfectly fine. You hardly notice anything about them. You'd have to age them out a really long time and look really hard before you saw any defect in those animals. You give them two bad copies and now they die before they're even born. And you have to work your system around a little bit in order to develop the cystic disease in the mice. Now with a pig, if you have one bad copy of the gene, then you get PKD. So the pig is behaving more like the mouse. Now, there are different ideas about why that is. It may be related to the way that the gene is expressed and produced. It could be more about the physiology of the animal. The physiology is just kind of what goes on the inside of it. For rats, Lisa, correct me, a mutant rat, it's autosomal dominant, is it not?
So I'm thinking about the RC rat. No, I think it's autosomal recessive
Okay.
because it's in the rodent class. And
maybe
the critical
RC
thing, the critical issue, Steve, I'm drawing on my memory cells here, but I think the pig has those pseudogenes.
I don't think the pig has the pseudogenes, but it does have, I think, the quadruplex, like Greg Van Hoovel spoke of last week. But we're kind of getting into the sticks and weeds of things there. But
yeah, I'm sorry, I didn't mean to send us all down this rabbit hole.
You had also talked about the expense, though, and just to give you an idea, we pay for our animals what's called the per diem, and our per diems for a cage of 5 mice is about $1.50 per day that we pay to manage their food, their changing, their vet care. The per diem on a single pig is probably on the order of $50 or $60 a day. So that cost is really prohibitive towards university research, unless you have a really, really big grant.
Yep. Thank you.
Yes, you're right about the quadruplex, Steve, now that I'm thinking about it.
What kind of grant you're thinking for doing more with the pigs or the animal that is more closer to human being rather than a mice?
What I'm not working on any grants with the pig. I think you would probably have to have, I don't know, Lisa, what do you think? Close to $750,000 to do a decent study
Yeah.
with a pig model.
And
Most
and
NIH grants are about $250,000, but you probably need something about 3 times larger than that. So there's just not a good mechanism to fund those types of studies.
right. And you know, you can do it in academia if you have other sources than the NIH. And you know, and you know, so I was shaking my head when Steve talked about what it costs to take care of mice and keep mice to do these kinds of experiments at KU versus what I remember at Children's National, what I know is true at CHOP. It's quite different. And so, you know, I think, Teslima, one of the things that's really important is to be really thoughtful about the experiments that you're designing so that they really match the model that you're using, so that you can generate biologically important data. Trying to do things in the mouse that really should be done in the pig is going to be a prescription for disaster. So matching your resources to the model that you can use as you design your experiments is important. And that's a reality, but it's also part of the richness of the field. I mean, this is why people have used experimental models that, you know, like, for example, zebrafish, that they don't really have a kidney, but they have a framework that if you ask the right questions, you can use to advance biologic knowledge of a much more complicated structure like a kidney.
So could I ask another question that's just related to that, unless there is a time limit? Is that okay, Reem?
Sure, Taslima, go ahead. That will be our last question.
Okay. Then the question is that you cannot replicate this mouse study with the human being, right? And so this is the mice you do, then you have to produce it with some kind of to go into the human beings, because like you know, there are many kinds of differences between the human being and the mice. So question is that what would be the next step after the mice? So where you found the like this little animals and going to the human being, what is the next step will be?
Yeah, it's a little bit more of a clinical question. At least I'll let you tackle that one.
Yeah, it's such a good question, Teslima. And it's the question about animal models, right? Because I mean, it doesn't take a rocket scientist to realize a mouse is not a human being. And so you can't approach this with an impatience that this is going to be a perfect model. Let me take, I hope, a careful step in a slightly different direction to answer your question. So there is there, you read in the press that in the current sort of leadership of Health and Human Services in the current administration, that they want to do away with animal models because their point is mice are not people. We should be working on people, or we should be using AI, or we should be using cell model systems, or we should be using, these, you've heard of iPS cells and these kinds of, we should be doing all these kinds of things. The problem is they're all approximations. And so, if you will, I'm going to switch from cars to eggs. You can't put all your eggs in one basket. And part of the reason the field has moved forward is because there are people like Steve and to some extent me who work in mice. There are people who work in rats. There are people who work in zebrafish. There are people who work in cell lines. And that knowledge, one of the things that we do is we make an insight in one system, and then we say, is this real? And so we test it in other systems. And even then, the translating that knowledge into humans is not 100% foolproof. And so it's a constantly iterative process of using these models and these systems to come up with an insight that you can test in humans. And so it's not sort of, again, it's not a straight, path. You do cells, then you do mice, then you do treatments in mice, and then you go to humans, and bingo, there you have it. So when people talk about, because you could ask the question, well, why are we working in mice and in rats and in pigs and in zebrafish and in cell lines? It just is too much. Why don't we just pick something? Picking something would foreclose our ability to gather the knowledge that we need. And you have to be committed to being able to, to know when we're not going anywhere with this. We got to have a different line of inquiry. And so it's a constant, kind of 2 steps forward, 1 step back, readjusting to get to that scenario where we can test things in humans. That's with a discovery of a new insight. Meanwhile, back on the ranch, biology is biology. Remember, tolvaptan was not developed as a PKD drug.
Yes, yeah.
It is a drug that hits a pathway that Vince Torres and a whole lot of other people said is important in PKD. So meanwhile, while we're doing all this targeted, or we're coming up with these great ideas and testing them in our various models, Steve's absolutely right. He made a point as kind of an aside, but I think it's really important. Really good scientists and really good resource groups like ours are very mindful of what else is going on in science and in the literature. And people knew that tolvaptan being developed for, a very different set of disorders hits this pathway. We should try it. And again, where they tried it was in experimental models. So there are multiple different directions. And this is why, as we think about where we are right now, we need to be vocal about the importance of this multifaceted approach to science, because we're not smart enough to know where the pay dirt is.
Yeah, that's why, my earlier questions that how long, a researcher like Stephen or Lisa, you can take. So the answer I got that time, it won't be 20 some years, but now I think it might be more than 25 years to get into the like a human being, because
it could be.
Yeah, so that's why, that's the reason I asked that.
Right, but I think that the other possibility, again, just to go back to something that we said earlier, is, so I think there's an expression, Steve, correct me if I'm wrong, about cars firing on all cylinders to make a car go really fast.
Yep.
And to make the science go fast, we have to fire on all of our cylinders, all of these different sorts of things. And like Fleming, who was a sloppy scientist, but he was very observant, keep paying attention to what we see. Because, Tesla, it could be 25 years. No question it could be 25 years. But it could be much sooner if people doing these kinds of experiments, or even experiments in another field, make an observation and go, huh, that could be related.
That's what my point also, that, it is, as you say, that the tolvaptan was not for the PKD, it was for something, and then someone thought, okay, it can be used for PKD.
Right.
So similar kind of things, what Stephen has described so nicely, and especially with the analogy of the car. But to me, it seems that it is going to take, like in the long, the road is not very close. They're looking at the summit from far distance. So.
But I'm 52 and I hope to be pushed out of this field before retirement does. I hope to be pushed out by effective therapies and effective cure, and there won't be any more funding for me in this field, and I'll have to find something else to do with that's my hope.
No,
Well,
definitely, yeah.
Okay.
The reason I asked the question that.
Yeah.
Okay.
Thank you, Steve, for all your optimism. It's really nice to hear.
Yeah.
Thank you very much for your talk.
Yeah.
Absolutely.
It was a great
We're
talk.
5 minutes after. Steve, I sincerely think this is the best talk I've heard about basic science in PKD or outside that truly brings it to the ideas and the principles. So we can't thank you enough. I think this was the perfect subgroup to start communication with our patients, but you set the bar so high. We'll see how your colleagues do.
Reem, you should share his slides and the ideas with the other folks so they understand.
Oh, believe me, that's why I'm like, okay, let's make sure we're recording. This is going so well. Okay. I believe, thank you so much, Steve. I appreciate it. I believe, Nicole, that we might have another scheduled meeting on next week. Is that correct?
Next week, our next meeting is supposed to be next month.
Yes, but there has been...
I'll be sharing the information with the next. So I'm working with the other subcommittee heads and getting the next meeting scheduled.
Okay.
Oh,
so,
I didn't know. Okay.
We'll reach out to everybody, make sure it's a time that works for everybody, but we're going to have more of these meetings. And again, thank you all. This was wonderful. Steve, thanks.
Yeah, thank you all.